While we are waiting for students to log in, let me say that, let me tell you a few things. I've got some grading done and I'm calling it up now so I'll remember the numbers. I know I got one old lab done, meaning graded it for the second time. And that was lab 5. So lab 5 is now graded for the second time. And I've got lab 8 graded for the first time. So take a look at those. Let me make sure that I made it so you can make corrections to lab 8. Let me just look at that now. Nope, I got to do that. I forgot to do that last night. Sorry. Let me get that done now. So that'll be due, the new corrections will be due this Saturday. And that'll be the 13th of November. Yeah, I'll just change that now. I also got quiz 3A, I think it was, graded. And so take a look at that. Oh, that's B. Yeah, I got quiz 3A graded. And let me see if I have answers up. If I don't have answers up, I'll get those up now. Week 5, maybe. Week 6. Okay, the answers are already up. So quiz 3 answers are up. Quiz 3B should show you the answers for a week after you take the exam. Well, it's 2 days after you take the exam, I show the answers, and then they're up for a week. But quiz 3A is up now because I just graded that. Any questions about any of that? Let's turn to the syllabus. And I gotta shut down the grade book. And then I'll turn this on so you can see it. All right, so showing my screen now. There's the answers, quiz 3A answers are right underneath quiz 3A. Come on. So today is October 9th, and we should be on chapter, and hopefully we'll finish that today. Is there a question?
It's November.
Oh, sorry, November 8th. Thank you. Yes, November 8th. It looks like on Thursday there will be no class because of Veterans Day. We are having a lab today at the normal time is at 6:30. Drawing a blank here, 6:30, lab 9. And then there will be no lab on Thursday as well. All right, any questions about any of that? We should finish chapter 8 today. I don't know if we're going to succeed. We're a little behind schedule. Any questions on any of that? All right, if there's no questions, let's begin. I think we went further than that. Ah, yeah, we talked about mutations. So we had talked about missense mutation, and that's a change in one base, and it results in a change in the amino acid. Unlike a silent mutation, which changes the DNA, but does not change the protein, the amino acid in the protein. There's also a nonsense mutation, That's where you're changing one base, and instead of the codon then coding for an amino acid, it codes for the stop codon. And so this is a nonsense mutation. Like when you change, sorry, I'm having a hard time reading this. Let me get it a little bigger. U coding for the amino acid phenylalanine to UAG, it codes for stop. That means you'll make the protein up to this stop, and then you'll have a truncated protein, a smaller protein. That protein may lose its function. There are some cases where this was seen, like some cases of muscular dystrophy and cystic fibrosis patients. The individuals have UAG coded or a nonsense mutation in their DNA for coding for a stop codon. There's also a frame shift mutation, and that's where you add a certain nucleotide to a DNA, or you take out a nucleotide from the DNA. So there's a DNA base that's missing or added, and it can be more than one nucleotide. It's a frame shift mutation as long as it's not a multiple of 3. If you pull out 3 nucleotide bases, what you do is you pull out one codon, and so you'll get a different protein. It'll be missing one amino acid, but it's not a frame shift mutation because you pulled out a multiple of 3, or you add a multiple of 3. A frame shift mutation is where you change the nucleotides by, let's say, adding 1. And I've got that shown here, where we have U, and then we remove one nucleotide, the U, which is shown here. Let me see if I can scroll that over. Just move it over. Oh, didn't want to do that. That's good enough. I need to get this out of the way. So instead of having U, we then pull the G from the next codon, and then we'll have UUG, which instead of coding for the amino acid phenylalanine, will code for the amino acid leucine. However, now instead of having GGC coding for the amino acid glycine, and then UAA coding for the stop codon, we then will have, sorry, I'm losing my paces here. We'll then have GCU, instead of coding for glycine, we'll code for alanine. And then we'll have, I don't know, UAA coding for another codon, coding for another amino acid, and it's not the stop codon. And then this will continue adding more amino acids until we reach another stop codon, or the messenger RNA ends, in which case you won't get another amino acid. But you see how this frame shift mutation, from this point here, where the frame shift mutation is, all of the amino acids in the protein after that are altered. And that's why it's called the frame shift mutation. Any question about any of that? Some examples of a frame shift mutation are some Tay-Sachs patients, where they have an extra nucleotide base added or a nucleotide base removed, resulting in a frame shift. Any question about the different types of mutations? All right. Mutations can be of 2 types. There can be spontaneous mutations, and that's simply where, when the DNA is replicating, the polymerase makes a mistake and puts in the wrong nucleotide. or I guess in a case of a frame shift mutation, a nucleotide doesn't get added. And then there are spontaneous mutations, and these occur by mutagens, such as two types, radiation and chemical mutagens. Any question about any of that? We have two types of radiation which can cause mutations. There's ionizing radiation, and that includes X-rays and gamma rays. These are radiation with a high amount of energy, and when they hit an atom or molecule, they will form ions. They give the electrons the energy they need to leave the atom. And then these ions can react with DNA. If the DNA is not repaired, it will cause a mutation. So ionizing radiation hits this atom, gives the electron the energy it needs for the electron to leave the atom, and then that makes an ion, which can then go bump around in the cell, changing molecules, including changing DNA, which can lead to a mutation. There's also UV radiation, which works in a different way. UV radiation, when it hits DNA, it causes the thymines to form a covalent bond between them. I'm talking about an extra covalent bond, actually two extra covalent bonds. So besides being linked together in the phosphate ribose linkage, they will have two covalent links between the thymine, and we call that a thymine dimer. These thymines then will not link, hydrogen bond, I should say, to their corresponding corollary nucleotides, the adenines. So there will be no hydrogen bonding right here. And this creates a bubble in the DNA, meaning the DNA isn't quite right. And that bubble will stop the DNA from being replicated or from being transcribed, meaning the polymerase can't read past the thymine dimer. This has to be repaired or it will lead to a mutation. We're not going to talk about the repair. We're not covering light repair because humans and placental mammals do not have light repair. All right, let's talk about a chemical mutagen. This is showing you what happens with nitrous acid, sorry. Nitrous acid is a chemical that acts as a mutagen. And when the nitrous acid reacts with DNA, it can change the adenine. So it's still adenine, but it's changed. And now this adenine does not bind, hydrogen bind with the thymine. And worse, the polymerase, when it sees this adenine, it doesn't see it as an adenine, it'll see it as a guanine. And so when the polymerase replicates this strand of DNA with the altered adenine, instead of putting a T over here on the newly replicated strand of DNA, it'll put a C. And then when this strand or this strand gets replicated again, we have fixed that mutation. The GC nucleotide base is now coded in the genome, and you cannot go backwards, back to the AT. At this point, right here, the cell could recognize that this is something wrong here. And if it realizes that the error is with the A, of course, the C also needs to be removed. So that wouldn't work because they wouldn't know what to do. You got to catch it, the error right here. If you catch the error here and get this A replaced and put in a new A, you can correct that error. But right here, even if you remove this A here, it'll see the C and then put a G over here. And so that'll fix the mutation. All right, any question about any of that? When we're talking about a mutation, spontaneous mutation happens about once in every 10 to the 9th replicated bases. Now, a gene has on average about 1,000 bases. And so that means the chance of a mutation in a gene is about 1 in 10 to the 6th, or 1 in a million. If you happen to know the human population, it's something like, is it 7 or 8 billion? Something like that. You can see that children born, every once in a while, we're going to have a child that has a mutation in a gene. Okay, any question about that? Mutagens simply increase the mutation rate over and above the mutation of spontaneous mutation. And they increase the mutation about 10 to 1,000-fold, depending on what the mutagen is. And so that means the chance of a gene being mutated when exposed to a mutagen is 1 to 10 to the 5th, or as high as 1 to 10 to the 3, meaning 1 in 1,000 genes could be mutated. All right, any questions about mutations? If not, let's move about talking about how we identify a mutant. You can identify mutants experimentally in 2 ways. You can use a positive or direct selection, or you can use a negative indirect selection. This here, if you saw somebody walking down the street looking like that, you'd say, oh, there's a mutant. And that's using positive direct selection. You can detect that that individual is a mutant because the cells or the individual looks different, appears different, grows different, or acts different. So there's something that is different about them, and you can tell that they are a mutant. A negative indirect selection, you detect that there are mutant cells because they do not grow. And let me show you negative selection. I did this when I was in college. You can look for negative mutation in bacteria or in yeast by growing the colonies on a petri dish. And then you get a velvet stamp and you press that stamp onto the petri dish. The cells from the colony get placed on the stamp. And then you use this stamp to stamp 2 other petri dishes. This petri dish contains the medium with the amino acid histidine, and this petri dish contains a media lacking the amino acid histidine. You then let the cells grow up, and you'll see that wherever there was a stamp here, you'll get a colony, which pertains to, I think that's that one there. And then you see this colony here grew on the media with the amino acid histidine, but on this plate, lacking the amino acid histidine, no colony grew. These cells are mutated and they will not grow unless you supply the amino acid histidine. It may be that the mutation is in the synthesis of the amino acid histidine. And without it, the cells won't grow. anyways, that's negative selection. Any question about that? All right, let's turn to genetic recombination. Recombination is the exchange of genes between two DNA molecules. It can happen with crossing over. Crossing over is where two chromosomes come together, and this happens in meiosis. And you'll notice one chromosome, we'll say it's from the father, has all lowercase alleles. The chromosome from the mother has all uppercase alleles. The two chromosomes can cross over and then break right here. And then this portion of a chromosome can join that chromosome, the father's chromosome. And then this portion of the father's chromosome can join the mother's chromosome. This is called crossing over, where now we have the mother's chromosome with lowercase e, f, and g, and then the chromosome has the alleles from the father's chromosome, capital H, capital I. This is crossing over. What do you notice between crossing over and recombination? Are they the same thing, something slightly different, or something totally different?
Slightly different.
Yes, they're slightly different. And that is, recombination is a little more general term. You're just talking about genes moving around from one location to another. And then crossing over is the physical breaking of two chromosomes and then exchanging parts of those chromosomes. So whenever you have crossing over occur, you have recombination occur, but recombination does not have to happen with crossing over. It's a little more general of a term. For example, you could have recombination happening like when a piece of DNA is brought into the cell and it then recombines with the DNA and replaces the DNA. That occurs, that recombination event occurred without crossing over happening. All right, the point of recombination is this does not change the genetic information, it just reshuffles the deck, meaning it makes the genes come together with other genes, but it does not add new genes. The only way you can add a brand new gene is by mutation. And I mentioned that this happened once in the human population about 10,000 years ago, where we had a spontaneous mutation, and now humans have the genes for blue eyes and for brown eyes. Before, we only had the gene for brown eyes. Maybe sometime in the future, we'll have another mutation and humans will get the yellow eye gene. I don't know if it's possible with humans, but I know dogs have yellow eyes. And so it's likely that humans could have yellow eyes as well. All right, so we mentioned that the only way to get a new gene in a population is with spontaneous mutation. However, there are ways of getting a new gene in a cell or in an individual in a population, and that is with horizontal gene transfer. Before I talk about horizontal gene transfer, let me remind you that vertical gene transfer is the movement of DNA or the information in DNA during reproduction from parent cell to daughter cells or from parent to offspring. That is vertical gene transfer, and it's the transfer of information between generations of cells. Horizontal gene transfer is the transfer of genes between cells of the same generation. So you have one cell over here, and a gene from it can go into another cell. And that's horizontal gene transfer. Horizontal gene transfer can happen in 3 ways. It can happen with transformation, conjugation, and transduction. Let's talk about each of those 3 ways. Transformation is where DNA comes out of a cell and is naked DNA. Probably it is coming out of a dead cell. That naked DNA can then be taken up by a recipient cell. And then a portion of that naked DNA taken up by the recipient cell can recombine with the chromosome and then be expressed by the cell. So, oops, went too far. So the new genetic information coming into the cell is lowercase a, b, c, and d. Only the lowercase a gene gets incorporated into the chromosome, and then the lowercase a gene is expressed, and that is a new gene for this cell. The other part of the DNA that's brought into the cell will, in this case, be degraded, and then the nucleotides can be used by the cell. This is transformation, where the lowercase a gene comes out of the cell into the cell, and then is expressed by this cell. So this cell can pick up a new phenotype, such as if lowercase a gene codes for drug resistance. The cell will now be drug resistant. So this is a little video about it. The cell sees some DNA, consumes it or brings that DNA into the cell, gives it indigestion, and then the cell changes its phenotype. Any questions? That is transformation. The cell will change its phenotypes if it picks up a new gene that it expresses. If it does add a gene it already has, then the cell will not be changed. All right, the second way that horizontal gene transfer can happen is by conjugation. And this occurs in gram negatives with the sex pilus. With gram positives, we can get conjugation. And what happens is 2 cells come together. And then the cell wall and the cytoplasm, or not the cytoplasm, the cell wall and the cell membrane open up. And then the cell membrane, let me try and blow that up a little bit. This cell binds to that cell. And so the cell membrane goes around and then around and then around and then around to here. And that means that the cytoplasm from this cell can exchange with that cell. When that happens, DNA from one cell can move into the other cell. And I'm not going to talk about that. You can read a little bit about it in your book. But the mating bridge is something that is not understood as well as the sex pilus and gram negatives. In gram negatives, you can have sexual conjugation happen where you have an F- cell, and it has a plasmid that codes for the sex pilus. The sex pilus is hollow, and it literally skewers an F+ cell. So this cell has a sex pilus. When it finds an F+ cell, it skewers it, and then the sex pilus can act as a winch and then bring the two cells together. They do not have to be right next to each other because the sex pilus is hollow, and the plasmid from this cell, once it's replicated, can move through the sex pilus and then go into the F plus cell, which is shown in this diagram. And in this diagram, you see that the F plus cell and the F minus cell are right next to each other. That doesn't have to be the case. They can have the sexual conjugation happen over this, but that sex pilus does act as a winch and pulls its two cells together. The F plasmid then replicates, and a copy of it moves from the F plus cell into the F minus cell through the sex pilus. Remember, the F plasmid codes for the sex pilus, so that this F minus cell, once it gains this plasmid, now will start to grow a sex pilus. And so we say this cell has changed from an F minus cell to an F plus cell. And now this cell can go and find an F minus cell and skewer it with its X pilus after it grows it, and then copy this plasmid and send it into the F minus cell. Any question about any of that? All right, the third way that genes can move around in a population by horizontal gene transfer is by transduction. Transduction uses a virus, and we haven't talked about viruses yet. So this is going to be sort of an introduction for you with viruses. What happens is a cell gets infected with a virus, and then the virus replicates and will lyse this cell. I thought I had a better picture than that. Let me shrink that and see if I'm missing part of that. Nope. All right. We'll talk about it later. Exactly. But the virus, when it replicates, let me blow this really big so you can see the color here. It degrades the host chromosome, which has a bluish purple color. And why it degrades the host chromosome in this cell is the virus DNA wants to call the shots and run the cell. And an easy way to take control of the cell is to degrade the host chromosome. And then only your genome is running the cell. And then what the virus does is it replicates itself, replicates its viral DNA, replicates its viral protein, and then makes new viruses, lyses the cell to shed the viruses. But as you see, mistakes get made. And this virus here is correct. It has a reddish purple genome. That's the viral DNA. But this one is a blue purple DNA. That is the DNA from the chromosome of this cell. And then when this virus goes over to infect this cell, it normally will bind to the cell and then put its genome into the cell. But instead of putting the viral DNA into this cell, what it's doing is it's putting the DNA from this cell here into the cell. That will give this recipient cell new DNA from this cell. And then this DNA can incorporate into the chromosome of this cell and then be expressed. And that can give this cell new genetic information, DNA that was once in this cell. For example, if this cell is sensitive to antibiotics like chloramphenicol, and we have drug resistance to chloramphenicol on this DNA coming from the virus, and then that viral DNA is incorporated into this chromosome, this cell will now be resistant to the antibiotic. Any question about any of that? So those are the three ways that horizontal gene transfer can happen, where cells can pick up new DNA from another cell in the population. And that can happen by transformation, by conjugation, or transduction. The DNA has to exist someplace in the population in order for the cell to pick up this new genetic information. Because if it doesn't exist anywhere in the population, the only way the cell can get it can't get it from horizontal gene transfer. The only way it can get it is by spontaneous mutation. And I mentioned that spontaneous mutation happens about 1 time in 10 to the 6 genes, or 1 in a million. How often do you think transformation happens in bacteria? Where the heck is that button for? Oh, no? Oh, there it is. Let's blow that up. Nobody's taking a guess? You think it's more or less than 1 times 10 to the 6? So spontaneous mutation, we just got to SM for that, is equal to 1 times 10 to the 6. Transformation, why is that so little? Oh, I know why, I got it on that. Come on. Nobody's going to take a guess. Transformation in bacteria happens about 1 in 10 to the 5th cells, or about 10 times more common. Conjugation happens about 1 in 10 to the 5th, or about 10 times more common than spontaneous mutation. And then transduction, I got too many T's here. Oops. Also happens about 1 in 10 to the 5th cells. So all of these horizontal gene transfers are happening about 10 times more often than spontaneous mutation. So they are very important to a population's evolution. Any question about any of that? Now humans have spontaneous mutation, right? Anyone even going to agree with me? Humans have spontaneous mutation. And in a sense, we have conjugation too. Sexual reproduction is the combining of 2 genetic information from the parent, creating a child, which will be different than both of the parents. Although it's obviously very different because we are a sexually reproducing species. And this is just adding a few genes by sexual conjugation. Humans do not readily have transformation. For example, my cells, if I put them on the table and then a cell falls on the table from my cell, somebody coming over to the table, they're not likely to pick up the DNA from that table. And then you would have to have it happening in a gene, not a gene cell, a gene, what's that called? Genome cell? No. Somatic cells and germline cells for it to be important for the population. And like I said, it really doesn't happen in humans. Transduction. Do you think transduction happens in people? Anyone gonna guess? Well, transduction happens with a virus. Do humans ever have viruses? Which can make them, huh?
Yes.
Yes, humans have viruses. Like for example, in our noses, we have the common cold. There could be all the time in our noses, our cells picking up DNA from somebody else's nose cell. So transduction probably does happen in people, but it doesn't happen in the germline cells. If it happens, it probably happens mostly in the nose and the throat. And that's not going to be important for the population. You have to have it hit the germline cells before it would be important for the human species. So humans mostly have spontaneous mutation. We don't have lateral gene transfer. Like I said, our nose cells do, but who cares if my nose cells pick up a gene from, I don't know, somebody in the Clark College. My nose cell doesn't care, and I'm not going to pass that on to the next generation. So it's not important for the population. All right, any question about horizontal gene transfer? If not, let's talk a little bit about plasmids. We introduced it a long time ago, I think in the beginning of chapter 8. Plasmids are pieces of DNA outside of the chromosome. They're small. In prokaryotic cells, plasmids are circular, but otherwise they're the same as the chromosome, except they're much smaller. Like a chromosome has like 100,000 or 200,000 genes on it. A plasmid only has maybe like a dozen genes on it. And here's a plasmid here showing you the different genes. And plasmids do replicate independently of the cell's chromosome. It doesn't need to replicate the same time as the chromosome. And we actually saw that. Let me go back a little. Right here, where this plasmid is replicating and the chromosome of the cell is not replicating. So plasmids can replicate independently of the cell. Any question about plasmids? Most eukaryotes do not have plasmids. It is true yeast do, but a lot of bacteria have plasmids. So they're more common in bacteria. I'm not going to say the majority of bacteria cells have plasmids, because the majority probably do not, but they do have plasmids. There are different categories of plasmids. All you need to know is there are different types of plasmids. You don't need to know the different types. Is that clear? So you don't need to know there's a conjugative plasmid or an R factor plasmid. You just need to know there are different types of plasmids. There's another piece of genetics we need to talk about, and that is a transposon. It turns out that a transposon is a region of DNA that moves around even more frequently than plasmids. And I guess I haven't mentioned that. A plasmid can move from one cell to another cell, and a plasmid can also rejoin with the chromosome or come out of the chromosome. So they can sort of move the DNA around. Well, a transposon is even more into moving the DNA around. A transposon is a segment of DNA that moves from one region of DNA to another. For example, in my cells, I might have a transposon on chromosome number 21, and it can move out of chromosome 21 and then move into chromosome 23. So the DNA moves from one location to another location. Transposons always have an insertion sequence where they have the transposase gene in the center and then inverted repeats on the ends. ATC, and the inverted repeat is AC. How does that go? We're just going to say it's an inverted repeat. It might be inverted from this one. Yeah, it's inverted from this one. It starts ACT going this way and then going this way, ACT. Inverted repeat. The transposase gene cuts the DNA right here and then reseals the chromosome. And then it will cut the DNA in another location. And then the inverted repeats help the DNA combine in the new location. And then it'll seal that new location. And that's what a transposon does. And that's a simple transposon that all it codes for is the transposase. You can have a complex transposon, which carries other genes in the transposon. So transposon 5, for example, carries the kanamyosin resistance gene. And it has actually a transposon right here and a transposon right there, because if you notice, there's a transposase gene, and there's the inverted repeat, and there's the inverted repeat, and we have that over here as well. Together, that will allow this to move out of one location and then move into another location. And the kanamyosin resistance gene can move out of the chromosome and then move into a plasmid. And then the plasmid can move out of this cell and then into another cell, bringing along the kanamyosin resistance gene. So you can see that transposons really help shuffle the DNA around in the cell, as well as between different cells. Okay. Transposons are found in eukaryotes. I'll just mention that here. That's where the transposon gene moved into a plasmid, and the plasmid can then go to another cell. Transposons were actually first discovered in corn, a eukaryote, and they noticed that the corn had the corn kernels of like an Indian corn were different colors, and that they were not breeding true. And they studied what was going on and found out that the corn colors, especially the ones that were in between solid, blue and solid red, that they were a mixture of blue and red together in the corn kernel, were happening because of transposons, where the genes in the cells were moving around from one location to another location. And humans have transposons, but to tell you the truth, I don't know anything about human transposons. All right, any question about transposons? All right, let's talk about our last slide, genes and evolution. evolution happens when a population has genetic variation, and all populations have genetic variation, and then different variants are selected by natural selection, and then that will result in the population changing that we call evolution. So as long as a population has different genes in the population, where different individuals or different cells could have different genes, we have genetic variation. Genetic variation first arises from spontaneous mutation. Crossing over can shuffle that new genetic variation so that new genes will be together in combination with each other. So they won't be selected as a unit. For example, the blue eye gene used to be linked to the blonde hair gene. Well, you can now have blonde hair and brown eyes, as well as blue eyes and brown hair, meaning they're no longer linked. And that was like 10,000 years ago when they were linked. And that happened simply by crossing over. Transformation, conjugation, and transduction can shuffle the genetic variation between different cells in a population, but it needs to first arise by spontaneous mutation. Transposition increases greatly the shuffling because it can move the genes around from one location to another and then into a plasmid, and that will increase the chance that it'll happen by transformation, conjugation, or transduction. And transposition sort of does the same thing as crossing over, moving the gene to a new location. Anyways, that creates the genetic variation in the population. And then selection will work, natural selection, on that genetic variation, changing the population, and that will drive evolution. Any questions about any of that? If not, we're done with the lesson. I thought we were going to be behind schedule. Let me not save that because I got to delete some things. All right, are there any questions? If not, I'm going to talk about our next chapter just a little bit. I think it's chapter 13, but let me check. Nope, chapter 14. Chapter 14, the principles of disease and epidemiology. I have, as always, an introductory slide giving you the major goals for this chapter, as well as a rough outline. In this chapter, I've only got 4 terms mentioned, but if you learn all the terms of this chapter, you'll learn about 50% of the chapter, meaning this chapter is very heavy on terms. And you can find the terms in the objectives for the chapter. It'll probably be in the study guide. There it is there. While I'm here, let me turn on chapter 8 so you guys can view that. If ever I don't have the link going for that when we're done with the lesson, send me an email as a reminder for me to get it done. I think I checked to make sure all the others were on earlier. Let me just double check. Yeah, chapter 6 is on. So Pathology, infection, and disease. Pathology is the study of disease. Etiology is the study of the cause of a disease. For example, the etiological agent for the disease anthrax is the bacterium Bacillus anthracis. And then pathogenesis is the development of a disease. Infection is the colonization of the body by a pathogen, or the presence of a microbe where it is not normally found. For example, all of us have E. coli in our intestines, so it is in our body. But if E. coli were growing elsewhere than the intestines, like in the ear, for example, it could cause an ear infection. And so E. coli being in the ear would be the presence where the microbe is not normally found. Disease is an abnormal state in which the body is not functioning normally. For example, if you have an ear infection, your ear may ache and you may not hear as well. You should come to understand that very few microorganisms are pathogenic. Remember, pathogenic means it's a microorganism that causes a disease. Like Bacillus anthracis causes anthrax. I mentioned that one because we've already talked about it, and Dr. Koch studied that bacterium to show that a microbe, specifically a bacterium, could cause a disease. And he used that study to make Koch's postulates. Few microorganisms are pathogenic, but some actually benefit the host. So we probably have more which are beneficial to us than we have that are harmful to us. The normal microbiota is mostly bacteria that is either neutral or beneficial to us. It is a large community of microbes found in association with all animals and plants. So all animals have a normal microbiota, and the human microbiota is specifically the human normal microbiota. You also have, like, for example, cat microbiota, and it can be different than human microbiota. For example, cats normally in their mouth have Bartonella henselae, a bacterium. I don't know what it does in cats, to tell you the truth, but it's in their mouth. And when the cat bites you, you can get cat scratch fever, meaning it causes that disease. So for us, it is a pathogen. And I don't know if it's a pathogen for cats or not. I don't know what Bartonella henselae does in cats, but it is part of their normal microbiota. I have a feeling, although I don't know this for a fact, we have Staphylococcus epidermidis in our skin, and my guess is that cats also have Staphylococcus epidermidis. So the species can have overlap, but there can be differences. Here's another one that's a different, and that is birds frequently have salmonella in their intestine, and it's part of their normal microbiota. Whereas people, if we get salmonella, it is a pathogen. The normal microbiota are microbes that permanently colonize their host. So our skin is normally permanently colonized by Staphylococcus epidermidis. The normal microbiota is only found in certain locations, certain regions of the body. The disruption of the normal microbiota and host balance is associated with diseases and with impaired immune function. For example, many of you have probably taken antibiotics, and you notice shortly after you took the antibiotics, you got diarrhea or constipation. That's because the normal microbiota host balance was disrupted, causing the disease. Usually the balance will reestablish shortly, and then the constipation or diarrhea will go away. Let's talk about the normal microbiota. One last slide, and that is it's only found in certain locations on and in the body. So all of the skin, all of the mucous membranes that are open to the outside, which is, I think, all of them, like the eyes, the mouth, the throat, the nose, the gastrointestinal tract, normal microbiota, and then the lower urinary reproductive tract, meaning the urethra in both males and females, has a normal microbiota. Places where we have normal microbiota, normal bacteria and other microbes growing all the time. Now, there are places in the body where we do not have normal microbiota. All of the internal regions like blood, cerebrospinal fluid, muscle, nervous tissue, and bone. I'm not talking about internal that's open to the outside, like in the vagina or the gastrointestinal tract. I'm talking about like your heart isn't supposed to have bacteria. If you do have bacteria in your heart, it is a disease, okay? So it's normally not found internally. You can have transient microbiota where the bacteria could be present on the individual for days, weeks, or months, but unlike the normal microbiota, the bacteria is there only transiently. The normal microbiota is there close to permanently, if not permanent, close enough. We'll have Staphylococcus epidermidis ever since we were born till we die. I'll give an example of a transient microbiota that I had. I don't think I've talked about it yet. I used to sample my skin every once in a while. You can just do it on a mannitol salt agar plate. And I was just looking to see if I had Staphylococcus aureus, because that is a human pathogen. And about 20% of Americans have Staphylococcus aureus growing on their skin. And I just wanted to see if I had Staphylococcus aureus. Okay, I didn't. And for years, I was just testing myself because I had the lab do it and I just showed the students how to do it. And I'd be testing myself for years. So about once every term for several years. And I never had Staphylococcus aureus. And then I went and visited my aunt, and she had a Staphylococcus aureus infection that she was receiving an antibiotic for to get rid of the Staphylococcus aureus. And she insisted on hugging me. And she always did that whenever we saw her. Although when she had the Staphylococcus aureus, I was a little bit hesitant. Anyway, she hugged me. And then the next time when I went and sampled my skin, I had Staphylococcus aureus on my skin for the first time. And I said, geez, I must have picked this up from my aunt. Fortunately, it was only a transient microbiota. And the next term, when I sampled my skin, I no longer had Staphylococcus aureus on my skin. And all future, all later terms, when I sampled my skin, I never had Staphylococcus aureus on my skin. So that was an example of a transient microbiota where I had Staphylococcus aureus on my skin for maybe a month or two. All right, any questions about any of that? Oh, I got two here already there. All right, if there's no questions, I'm going to end here and I'll see you at 6:30 for the lab.